Transgenic food, cure of diseases, design of animals for research. Genetic manipulation is real and it can be found in many areas of everyday life. Do you want to become Superman? What would happen if we design ecosystems at will? Do we want to live forever? Entering the personalization era. Genomic Cut&Paste, CRISPR-Cas9 system. DNA is a molecule that can be found in all cells of all living beings, although it's also present in viruses. It consists of a long sequence of 4 repeated letters, A, T, C, and G. Their multiple combinations lead to the information that is transmitted from generation to generation. This information explains how the organism behaves under different conditions, such as feeding, toxics, infections, susceptibility to disease, among many others. Genes are the smallest functional units of DNA, and the whole DNA sequence of an organism is called genome. Nowadays, knowing a gene sequence is relatively easy, and some gene functions are known. In fact, we have tools that can modify genetic information. One of the most extensively used tools is the CRISPR-Cas9 system. Roughly speaking, it detects a genetic sequence of an organism and then cuts it. Finally, the cut site is modified. In 1987, CRISPR sequences were found for the first time, but until March 2007, their natural function remained unknown. CRISPR sequences are part of a bacterial defense system against viruses that infect them. When a virus infects a bacterial cell, the bacterium incorporates part of the viral genetic material into its own genome. If the same virus infects the bacterium, it's recognized and its material is destroyed. Each integrated DNA fragment with viral information is called CRISPR due to its structure. It's like an internal library of all defeated enemies. So, if bacteria can incorporate new sequences to their DNA, why don't we use this mechanism to insert changes into an organism's genome? Then, CRISPR-Cas9 system has been artificially simplified for biotechnological use. Firstly, a gRNA string is synthesized in the lab. Mainly, RNA allows expression of the information stored in the DNA. However, gRNA is quite different to this standard conception of RNA because it's synthetic and it's used to detect the sequence we want to modify. In addition, a bound sequence must be detected. It's placed next to the target sequence. Then, an enzyme called Cas9 nuclease cuts the DNA string of the organism into the detected place. This allows modification, either by borrowing some letters of a gene in order to make it stop working, or by cutting on both sides of the gene. Finally, ends are joined. However, it's also possible to insert new sequences into the genome. This is the reason of the CRISPR revolution. For example, an unfunctional gene sequence can be replaced by its correct form. Also, new genes can be added and new functions are achieved. CRISPR-Cas9 gene editing system is an essential tool because it's specific, simple, user-friendly, cheap, fast, and it can be used in every life being. This mechanism is much more advantageous than former techniques and it allows progressing in many areas. For example, animal models have been created in order to study different diseases, such as mice with lung cancer, but this system can be used to cure diseases in those animal models too. In the United Kingdom, genetic manipulation of human embryos is permitted, but only in research. Genetically modified food can be more nutritive, plants can grow in hospitable climates, and their production can be increased, so more people will be provided. In the future, CRISPR-Cas9 could also modify whole ecosystems, for example, to eradicate the malaria mosquito. Opinions about this matter are extremely diverse and its acceptance depends on each individual. CRISPR-Cas9 can be beneficial, but it also opens a controversial debate. Aesthetic genomic engineering? Offspring design? Increasing lifespan? Food design for sybarites? Improvement of physical and intellectual capacities? All these questions can be discussed, but we must take into account the fact that CRISPR-Cas9 technology is a double-edged sword. What happens if we lose control of genetically modified organisms? What happens if it's used to create epidemics? And if we use it as a biological weapon against a race? What happens if changes are irreversible? CRISPR-Cas9 technology has great potential and it can lead to progress, but at the same time, it can trigger chaos. Modifying the information that tells us how we are maybe gives rise to an alteration of our essence, and maybe evolution course is altered too, or it's only accelerated. Future is in our hands. The use we make depends on us.